Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into medical school and other professional programs. And welcome back to MCAT Bites. Today we're exploring cell differentiation. This fascinating process is crucial for the development of complex multicellular organisms, allowing for a single fertilized egg to give rise to the diverse array of cell types in our bodies. Cell differentiation is the process by which a less specialized cell becomes a more specialized cell. It's a journey from totipotency to increasing degrees of specialization. Let's start with stem cells, the heroes and beginning of differentiation. There are several types we need to know for the MCAT. Beginning with totipotent. I'd like to remember this as they're totally potent, so they can become anything. They give rise to all cell types, including extra embryonic tissues. Only the zygote and early embryonic cells are totipotent. The pluripotent stem cells, I like to remember as nearly perfect with a P. They can differentiate into any cell type of the three germ layers: endoderm, mesoderm, ectoderm, but not extra embryonic tissues. Embryonic stem cells are pluripotent. And then we've got multipotent stem cells. These can differentiate into multiple but limited cell types. For example, hematopoietic stem cells can produce all blood cell types and white blood cells. And then we've got unipotent stem cells. These can only produce one cell type, but have the property of self-renewal. You'll often find these in muscle cell, gut epithelial, and lung cells, cells where we have a lot of turnover. Here's a question for you. What type of stem cells would you expect to find in adult bone marrow? The answer: multipotent hematopoietic stem cells, which can differentiate into all types of blood cells which we know are made in the bone marrow. Now let's dive into the mechanism of differentiation. At its core, differentiation involves changes in gene expression. While all cells in an organism contain all the same genetic material, different genes are activated or repressed in different cell types. This is controlled by various factors, first being transcription factors, proteins that bind to specific DNA sequences to control gene expression. For example, MyoD is a master regulator for muscle cell differentiation. We also have epigenetic modifications. These are changes that affect gene expression without altering the DNA sequence. These include DNA methylation, which often silence genes, histone modifications, which can activate or repress genes, and chromatin remodeling, which alters DNA accessibility. Finally, we've got cell signaling molecules, external factors that can trigger differentiation. For example, retinoic acid plays a crucial role in neuronal differentiation. The process of differentiation is typically unidirectional in mammals. Once a cell has differentiated, it can't usually revert to a less specialized state or change into a different cell type. However, recent research into induced pluripotent stem cells, or iPSCs, are challenging this notion and winning Nobel Prizes. iPSCs are adult cells that have been reprogrammed to a pluripotent state by introducing specific transcription factors. This groundbreaking discovery earned Shinya Yamanaka the Nobel Prize in 2012. Here's another question. What ethical advantage do iPSCs have over embryonic stem cells? Well, iPSCs can be created without destroying embryos, avoiding many of the ethical concerns associated with embryonic stem cell research. Differentiation doesn't happen in isolation. It's influenced by the cell's microenvironment or niche. This includes cell-to-cell interactions, extracellular matrix composition, mechanical forces, chemical gradients, and a bunch of other things we don't even know yet. For example, in a developing embryo, concentration gradients of morphogens like sonic hedgehog play a crucial role in determining cell fate. The balance between differentiation and stem cell maintenance is crucial for proper development and tissue homeostasis. Disruptions in these processes can lead to various diseases, including cancer. Understanding these processes is fundamental to developmental biology, regenerative medicine, and cancer research. Even more importantly, the biochem section of the MCAT. So definitely make sure you take good notes, grab some screenshots, put it into your Anki deck. And thank you so much for watching our mechanisms of cell differentiation, and I will see you next time.